All right, thanks for coming. Really excited about tonight's talk. My name is Robert Bia. If you don't know who I am already. And if you're new to the THS, this is our logo and mission statement. We are here in Sonora Desert of Tucson, Arizona, traditional homelands of the Tohono people and many other indigenous groups. I work at the Desert Laboratory on Tumamoc Hill. Tumamoc Hill is a transliteration of Chimamaik Doak, which means Horned Lizard Hill. And across the valley from Tumamoc Hill is the Catalina Mountains, which is called in O'odham Babat Doak, or Frog Toad Mountain, because of many lot of similar geological formations in the Catalinas, like the west side of Push Peak here, is very reminiscent of a giant toad. This is the cover of our most recent edition of Sonora and Herpetologist. It's a great newsletter that is free for reading on our website. And it commemorates Roger Rep, who passed away last November. We will have a celebration of Life for Roger on April 18th. So if you need more information, just get with me after the presentation. So we have a number of outreach opportunities throughout the year. We have already done SARSAF and Tucson Festival of Books. Coming up in August is the Southwestern Partners in Amphibian and Reptile Conservation. And in May, Sonora's first herpetological association just formed and is having a congress on the 21st through the 23rd of May. Again, contact me for more information. In July, we have the 50th International Herpetological Symposium, which is more focused on herpetoculture or captive breeding and conservation through that. And then the first weekend of October, the Tucson Reptile and Amphibian Show and Sale. And Ryan, we're going to possibly be at a reptile show in Phoenix on April 18th as well. Okay, yeah, get with Ryan for more information. This hasn't happened yet, okay. Volunteers are needed for this restoration project. So there's more information at the QR code. It's not moving forward. There we go. Okay. So these, there are two companion books to the Snakes of Arizona book. And they are currently being produced. If you have any information like photos or records that you'd like to contribute to the effort, you can go to this website and get more information on how to do that. I think the battery might be. So, that. Right. Anyways, that was a slide, so I'm trying to show you what. Okay, there we go. These are the upcoming speakers after this one. And tonight we have Taggert Butterfield, who's a good pal. I met Taggert through the turtle conservation world. He happens to work with one of my favorite turtle species, the spotted box turtle. But recently he has started working with endemic sliders. Well, at least one of them, the Yaqui slider, is endemic to the Sonora Desert region and the Río Fuerte slider. He's also a former student of Daniel Beck, who is a professor. Is he still a professor? Or did he
He retired.
retire? He retired from Central Washington University, and he was one of the first members of the THS and the first, he started the first student chapter. So that's pretty cool. He also designed our famous dancing Gila monster shirt, which Ryan is wearing here today. Great. Well, thank you so much, Taggert, and we will get your talk up and going. I'm just going to check with folks online and make sure there's no comments. They should be able to hear everything. And you might have to use this here to toggle or not. But if this doesn't work,
As a cycle.
Okay, well, thank you very much. My name's Taggert Butterfield. I'm the scientific director of the Mexican nonprofit called Students Conserving Nature, and we are dedicated to monitoring and conserving freshwater and terrestrial turtles. We work with 12 species, 7 in Sonora. I live in Alamo, Sonora, and it is the hotspot of turtles. There are 7 species in the area with several boundaries of different species. If you go on one side of town, you get one of the species. If you go to the other side of town, you'll get the other a different species. And it's an exciting place to live. We also live in Yucatan, and we also work in Yucatan. We don't live there. And Jalisco. And since everyone's tired of hearing me talk about box turtles, today we're going to talk about the work we've been doing to study and conservation concerns of Trachemys nebulosa and Trachemys yaqui. So the Trachemys genus is most diverse in Mexico. There's about 17 species of Trachemys and 28 or 9 subspecies, and most of those are in Mexico. And their taxonomy is constantly in flux, so the number of species and what are considered species can change, such as one of the species, Trachemys taylori, which is endemic to the Cuatro Ciénegas Basin in Coahuila. A recent study found this study actually suggests that this should be part of the Trachemys venusta group and should be considered its own species. However, we don't care that much about the species. We focus mainly on the populations. And in Sonora, we have 2 sliders, and these 2 sliders don't have taxonomic ambiguity. We know that they're distinct species. Although the Baja and Río Fuerte slider, these 2 subspecies are a little bit ambiguous, and there's doubt whether or not they consider subspecies. And then there's the Yaqui slider, Trachemys yaqui, that both occur in Sonora, with the Trachemys nebulosa also occurring in the Baja California Peninsula. So the Baja or Rio Fuerte slider has 2 recognized subspecies with weak genetic support for the subspecies, as I previously mentioned. And something interesting about this species is that a recent paper found that they have an alien mitogenome. They have a really difficult genome to be able to conduct phylogenetic studies with to understand the rest of Trachemys. And he calls it a genetic poltergeist, and hypothesizes that it probably bred or hybridized in the past and retained that genome. And when they run the genetics with mitochondrial and nuclear DNA, they actually come out sister to the diamondback terrapin, which is a marsh turtle that's on the eastern coast of the U.S. And there's debate whether or not this species originated in Baja California or on the mainland. But sound just became choppy. Stop that. Where did I get my sound? There. Oh, maybe I should talk over here then.
Well, there's microphones all along there.
Yeah.
Okay. Yeah. So although there is some doubt in the past literature, if you read, there's a lot of authors talking about whether it originated in the Baja California Peninsula or the mainland. But in our opinion, there shouldn't be any doubt whether or not it originated on the peninsula or the mainland. Compared to all other Trachemys species, Trachemys nebulosa really sticks out like a sore thumb. They don't have the typical, really bright colors of other Trachemys species. And as you go from north to south, from northern Sonora down to Jalisco, you have Trachemys yaqui and Trachemys ornata, and this kind of represents the continuum of Trachemys that we see, for example, on the East Coast. we're trying to divide those species is trying to divide up a color spectrum. You know, there's a continuous variation, whereas here there's a sore thumb in the middle of the distribution. And so, you know, this, although there's, you know, genetic evidence that they're very distinct, to me, it's much more likely they originated in the Baja California Peninsula and arrived to the mainland via reintroductions or migration. And for example, if you had, you know, 100 or 200 of these turtles in some bins, and you had to separate the Yaqui slider from the ornitis, these sliders apart, you'd probably make a lot of mistakes with Trachemys yaqui and Trachemys ornata because they look very similar, but you make no mistakes with Trachemys nebulosa because it looks so distinct compared to all other sliders. And in our opinion, they are the prettiest slider. So the Yaqui slider, you know, there's no taxonomic ambiguity. This is a slider that's actually described near Alamos in the tailwater of the Mucusari Dam of the Maya River, and it's distributed from southern to northern Sonora. Sister Trachemys ornata, I've already mentioned, and this is a picture of the type locality in the 70s when it was first described, and I went there and tried to take a picture, but there's this tree in the way.
It's a huge.
Yeah, and also one thing that can affect is that the dam flows. If you go there in April or during the summer, the dam's cut off and the river's going to be smaller. And if you go from now until mid-April, the river's pretty big. So maybe this patch of land shows up when they cut the dam flows off, but there is a ton more vegetation on the side now than there was before. So despite the wide distribution, there's almost nothing known about the ecology of these species. There's no peer-reviewed papers on their ecology, demographics, or abundance of any of the species. And information on populations is restricted to observations taken decades ago. And often when we're combing through the literature trying to find anything of what people observed in populations or how many they saw, we're often, you know, have to rely on single sentences like this, such as Trachemys nebulosa was easy to see from the mission lookout in Mulegé, which is in Baja California, in a book by Gismer in 2002. Defining information is like a scavenger. We have to go through the description of the species to see how many individuals they got, how they got them, and often they don't include that kind of information in descriptions or other literature. And when I started looking into this, I noticed that there's actually no peer-reviewed studies on any of the Trachemys populations in Mexico. There's been genetic work done, you know, a good amount of genetic work done on various species, but no studies on ecology, demographics, or population structure. And most of the information that does exist is in the Turtles of Mexico book, and a lot of it, you know, Richard Vogt worked for decades in Southeast Mexico, and he has information on all the populations, and he includes information about the males and the females and you know, anecdotal observations that he has from past and present of what he's seen, but no information on sampling effort and important information that we need to understand what's going on with these species now. And this lack of data to Trachemys is a direct threat to their conservation because it makes us unable to make a realistic conservation assessment, and it can lead us to believing that obviously all these turtles are doing good. We can focus on other ones when in fact they might not be doing so good. And in 2007, Trachemys yaqui was described or classified on the IUCN Red List as vulnerable. But this was based on no field data with geographic information systems and just a perception of what could be going on with the species, which might be, you know, a conservative designation to give it because, you know, perhaps it is vulnerable. We don't know anything about it. We know what's happening in the range, but it all might also create a barrier from other people wanting to study it. For example, if it was data efficient, as we recently described in 2023. You know, there's grants available for data deficient species, and it recognizes that really we don't have any information on the populations, which is an appropriate classification. And yeah, Trachemys nebulosa was evaluated in 2023. Both these evaluations are in press, and they also described it as vulnerable, but the people who described it had never seen these turtles in the wild, and it still can give us a false sense of security. Giving these kind of designations. And one important note on assessing conservation or doing conservation assessments of species is that they're based on population trends and life history traits. Just because, and the Central American river turtle is a really good example, because although the Central American river turtle, if we go to southern Mexico, Belize, or Guatemala, we can still find populations where we find a lot of individuals, and we go to those populations and say, this is doing great. And it has this wide distribution from Veracruz to Guatemala to the peninsula. So, this must be doing great. But what we know, since there was studies in the 80s and the 70s that looked at how many turtles were in the wild, and they're highly consumed, and there's also an observation that the populations in certain areas like Veracruz or Tabasco completely plummeted, it merited that this is a critically endangered species, because we know that populations have declined by more than 50%. So we can even go to populations today and find, there's a lot of turtles, but there might have been 4 times that amount 20 years ago. And with Trachemys, that's hard to assess because we don't have previous information. So assessing the conservation status of these species is urgent. They're large species. They weigh 3 to 6 kilograms, and they probably take 10 or more years to reach sexual maturity. And so if something happens in a population, it takes them a lot longer to recover in comparison with a species that can reproduce and get to adulthood in 5 years. And they depend on more perennial water sources. And one thing that people probably know about here in the southeast is that there's been an extreme drought in this area that's directly impacting a lot of species that depend on higher rainfalls or more perennial water sources. So our goal with these species has first been to establish long-term monitoring of their populations near Alamos, and we study these species in Alamos. I included the current distributions of these species on this map so you can see what it distributed. This is the type locality of Trachemys yaqui below the Macuspuri Dam. This is Alamos. This is where I live. And the main study site for Trachemys yaqui is down here south, actually south of their known distribution in a reservoir. Trachemys nebulosa, we've been sampling throughout the Rio Fuerte drainage and also this town south of Alamos. And then there's these ponds southeast of Alamos where we actually get both species. And we just discovered this recently, but it's pretty cool that we have this area where we get both species. And what we do, we assess the abundance, and how we're assessing abundance, how I'm going to be presenting abundance is using the trap effort per turtle, or the number of turtles trapped per trap night. And a trap night is how many traps multiplied by the number of nights. So if we put 4 traps, and that's for 2 nights, that's 8 trap nights. And we trap 8 turtles, that's 1 turtle per trap night. And if experts convene in a room to talk about the best methods to assess abundance, this would not be the best method. It's much better to have robust population estimates from year to year. But given the nature of getting these turtles and the amount of recaptures we get from one year to the next, it's the best estimate we have to assess abundance. We assess population structure and morphology, and evaluate threats of future viability by talking to locals, by knowing what's happening in the region, looking at precipitation, and observing what's happening in the habitats. So we're going to talk about Trachemys nebulosa first. In Sonora, we've sampled 5 sites from 2021 to today, or not today, but recently to the present. In Baja California, we got a grant to go to 4 sites throughout the peninsula. In 2023, in Sinaloa, we spent time visiting 11 sites throughout the Rio Fuerte drainage and its associated canals to sample the species in Sinaloa. So in Sonora, there's 3 sites that we sampled in the Cuchujaqui River. And one of these sites, John Legler sampled in 1961, and we do have tidbits of information of comparing past populations to present populations. I'll explain in a minute how we got those. There's 2 sites southeast of Alamos in artificial and natural reservoirs. This is a picture of one of these artificial reservoirs that was sampled. And in total, we've conducted 89 nights of sampling from 2021 to 2025, accumulating 200 trap nights, 36 unique Trachemys nebulosa, and 24 recaptures. So looking at the abundance, Trachemys nebulosa hiltoni in Sonora, the different sites that we've sampled. In the upstream site, we've traveled in the Cuchujaqui River. We've trapped 7 turtles and had 1 recapture. And these 2 sites combined, which are relatively close to each other, resulting in 0.16 turtles per trap night. In a midstream tributary, in a tributary that's about a quarter mile up from the Yaqui River, we've gotten 3 turtles, 3 recaptures, resulting in 0.2 turtles per trap night. And downstream in this site where that John Leather visited, we've captured 14 turtles and had 17 recaptures, which equates to 0.4 turtles per trap night. And the ponds southeast of Alamos, the ones where we find both species, we've got 3 turtles and 1 recapture, resulting in 0.14 turtles per trap night. And the lake in Piedras Blancas, the most southern site, we've got 8 turtles and 1 recapture, resulting in 0.71 turtles per trap night. And that's the only locality we've been able to trap juveniles. For some reason, in all turtle studies, juveniles are really evasive, but in this species, they've been especially hard to trap and see and figure out where they go and what they do. So I mentioned that John Legler sampled one of these sites in 1961. And so we can compare that site to now. However, in his book, in Legler and Vogt 2013, in the Turtles of Mexico, First, he did have data on about 30 individuals, but he didn't say how he got those, if he got those from locals, because we do know there's this famous local who lived in the region who would pay locals to bring them turtles. So we don't know if that's what he's doing or if he's trapping them. And he did know in 1978 when he went back after going in 1961 that there seemed to be fewer turtles and the area was more degraded with no details on sampling. And the area is now part of a natural protected area, and I would argue that's not all that degraded. It's still pretty well intact. And to figure out how they trapped and how many they trapped, I contacted the Natural History Museum of Utah and asked if they had any notes from Legler during these dates. And they actually found student notes because he brought students down as part of a class to do natural history work with turtles and other reptiles. And through these notes, I could figure out how many traps they put out and found out they trapped 12 turtles. And although they don't mention how many turtles they got on the last day they left when they took the traps out. But if we compare 1961, they got about 0.81 turtles per trap night compared to our effort over the past 4 years in the same spot where we got 0.4 turtles per trap night. So it does appear that there's less turtles in this site. So in Baja California, we sampled 4 sites for a total of 23 trap nights. We captured 9 unique Trachemys nebulosa and 3 non-native turtles. And we'll see those in a second. And these are a look at the sites that we sampled in the peninsula. We first went to Mulegé, and we only got 1 turtle, resulting in 0.17 turtles per trap night. And we also got an apparent Trachemys scripta hybrid, which I'll show in a second. And in the Purísima, we got 2 turtles for 0.33 turtles per trap night. La Higuerilla, we got 6 turtles, and that's actually to this date has been the best sampling success we've had in a couple of nights of sampling. In La Higuerilla, we got 2 turtles per trap night. And Presa de Buena Mujer, which Gismer in his book talks about the Presa de la Buena Mujer as a site with Trachemys nebulosa, and we tracked the Trachemys ornata. And one interesting thing about his book passage where he talks about this dam, so he noticed that fishermen were releasing turtles into the dam for consumption. However, when we went there, we only got that one ornata. Then one of the fishermen said, "Hey, I got some turtles from the dam at my house. You can come and measure them and look at them." We went to his house, and they were all Trachemys scripta elegans. So it appears that maybe Gismet in his book might not have known what species they're actually releasing into this dam. So we're not sure if Trachemys nebulosa are in this dam.
So elegans is the pet store turtle.
Yeah, Trachemys scripta elegans is the main pet store turtle, especially here in the U.S. It's a little turtle that only costs like a few dollars, and all the little kids see it and want it, and their parents always cave in. They definitely would have caved in to me if I would have pressured them, because I did cave in for a lot of reptiles. And so looking at the historical sites then versus now, which we basically have nothing. But Gismer 2002 commented that turtles were easily seen from the mission in Mulegé. And missions are those Spaniard churches that they put up all over the country. And there's this lookout there, and he mentioned from this lookout, they were easily seen from the lookout. And for example, in that Legler site, if you go there on any morning, throughout the year, you can see turtles there. They're basking in the surface of the water, and we also know how much our turtles are in there. So if there is turtles there, we should be able to see them from a place like this. But we did 3 observations there, and we didn't see any turtles. And when we trapped there, only 1 nebulosa was trapped, and we got this apparent nebulosa and scripta hybrid, which you can see it has the dark patterns, the dark coloration of Trachemys nebulosa, very little lines. It has the ocelli in the pattern instead of the lateral stripes that the scripta have, but it has a really red, distinct elegance mark on its head. And Purissima Roberts in 1982 observed 23 Trachemys nebulosa basking on rocks. And if anyone here sees this 23 nebulosa basking on rocks, please tell me where it is, because that was a really good population, and hopefully it still is, and we just weren't unlucky. But when we were there, we only trapped 2 individuals and also observed 2 other individuals basking near the surface of the water. So it does appear that there's less turtles in these sites. And in Sinaloa, we sampled 11 sites, accumulating 32 trap nights, and we trapped 10 Trachemys nebulosa. Looking at the sites, we started up here near El Macon reservoir, where the Río Fuerte comes out of the dam. We didn't get any turtles there. In La Misión, we only got 1 turtle. Down in Cuara, and then once we started noticing what the main drainage was like, and very little water, a lot of heavy fishing, we started going off into the canals to find where there was potentially pockets where we could get turtles. And in Cuara, we did get 4 turtles, resulting in 0.67 turtles per trap night. In El Charey, we didn't get any turtles. El Porvenir, closer to Los Mochis in Sinaloa, we got 4 turtles, resulting in 1.33 turtles per trap night. And in Higuera, Zaragoza, we got 1 turtle, resulting in 0.33 turtles per trap night. And that's one really hard thing about evaluating the conservation of a species, is that you have 2 or 3 weeks to go do the sampling, whereas having really concrete data takes years and a lot of effort in the same sites to really know what's going on. And one interesting thing for Sinaloa that we found out: every site we went to, every fisherman we talked to, every rancher we talked to, tractor drivers, all of them either eat or know of turtle eating, and they get them in their nets, and they eat them. Someone even told us that there was a restaurant that pays 50%, 50 pesos per kilo for a turtle. And that's really unique for northwest Mexico, because turtle consumption is really common in the southeast, in Veracruz, in Tabasco, in Chiapas, but it's really rare in the northwest. And so that was a unique observation of this area, seeming to be a lot of consumption of turtles in this area. And so looking at the population structure of these turtles, you can notice, looking at the distribution of the sizes, we found mostly adults. We don't have barely any individuals in these smaller age classes. And one observation is that females are bigger than males. It would be good to have a lot of information on juveniles and recruitment, and to really understand what's going on in these populations. But we basically just know that they're mainly comprised of adults, and females are bigger than males. In hiltoni, females average about 3.1 kilos, and males 1.6 kilos. In nebulosa, they're bigger. They average 3.6 kilos, and males 2.1 kilos. And big females are about 280 millimeters or 300 millimeters in nebulosa. And we've had females that have weighed 6 kilos, so they can get really big. And this is a picture of Trachemys nebulosa hiltoni, a male and a female from the Kentucky River, to show what males and females look like. So some conclusions and additional observations of Trachemys nebulosa is that they're not particularly abundant throughout the range. And the most abundant site was in La Higuerilla, where we got 2 turtles per trap night. And our anecdotal assessment, based on the tidbits of information we find in the literature, is that there does seem to be fewer turtles now than there was before. However, this isn't the kind of data that we can use to classify a turtle as endangered, potentially as near threatened, would be one of the classifications of the IUCN that we could give it. And the main threats, as I mentioned, Sinaloa, every site we visit, people consume turtles, and the non-native turtles present in 2 sites in Baja California, Trachemys scripta elegans and Trachemys farinata. And in Mexico, this really represents the gravest threat for Trachemys in the country. The Trachemys, they're the most diverse in Mexico, although we do have Trachemys scripta elegans, the greater slider that comes into the northeast of Mexico. Since not only Trachemys scripta elegans, but Trachemys venusta from southeast Mexico and Trachemys ornata from the Pacific coast are sold in massive quantities all over the country. If you go to Mexico City Market on any day, there's 3 or 4 big ones, and at each one you go and see a basket, with 1,000 little baby turtles in there that cost $1 a piece. And kids go them, they buy them, they put them in a bucket of water, and then when they can't take care of them, they go and release them. And in every major city in Mexico, basically, they're in the public ponds or canals they have. There's Trachemys scripta, venusta, ornata. There's a cesspool of released pets. And the problem with Trachemys is that they can hybridize with the native turtles, potentially threatening the genetic integrity of all these special Trachemys from Mexico. And this is an example of one for Puerto Vallarta, where we have a serious problem in a public lagoon, where we're trying to protect a critically endangered turtle, the Vallarta mud turtle. And you can tell this is a hybrid because one characteristic that is, all Trachemys elegans have, and none of the native Mexican species have, are these lateral lines on the costal scutes. And in Puerto Vallarta, there's this pond where people have gone to release these turtles. And not only do they go there to release them, they go there to take care of them. They're in love with the turtles. And if we take these turtles out, there'll be this huge political thing, and it'll come in the news that we're killing turtles. However, we know that most of them aren't supposed to be there, and we can tell that there's not only a ton of red-eared sliders, there's a ton of hybrids, which we can see. This one has ocelli like Trachemys ornata, but it also has these horizontal lines that are characteristic of Trachemys scripta. And in this Puerto Vallarta lagoon, there's 2 kilograms of hybrids and red-eared sliders for every kilogram of native turtle, and there's 3 species of native turtle. So they're really dominating this pond, and we've been working to come up with a solution on how we get rid of them. We can just take them all out and kill them, but then we get all the people in love with the turtles, who we also want to support, and we want them to love the turtles because we want people to conserve the other turtle there. And so, we're going to really have to take our time to get support from the community so they understand what's going on before we start taking turtles out of there, so we don't create a huge political scandal. And so moving on to Trachemys yaqui, so there's only been 2 sites so far that we've trapped, and we've accumulated 133 trap nights and trapped 67 Trachemys yaqui, so we've had much more success with this species trapping. And the abundance of Trachemys yaqui, and that the Presenta Taggiosa, which is an artificial reservoir south of their distribution, we've gotten 59 turtles, resulting in 0.54 turtles per trap night. So on average, over the past 40 years, we've had pretty good success at trapping these turtles there. At pond southeast of Alamos, which we just recently discovered, where they're found sympatric with nebulosa, we've got 8 turtles, and resulting in 0.3 turtles per trap night. And as I mentioned, they're sympatric with nebulosa, and this is, I said that we have, our nonprofit is dedicated to monitoring turtles and also developing students. This is our students in the boat there going to, or my wife and the students going out to set and take out traps in the Presa Taggiosa. So this is a look at the population structure and the distribution of carapace lengths. It's much more obvious that there's, females are bigger than males. The sexual size dimorphism is really clear, and since we have a lot of individuals, it comes up clear on the graph. And we have trapped 2 juveniles, one really small hatchling and also subadult. And females are slightly larger than Trachemys nebulosa hiltoni, and males are the same size as males in hiltoni. So females average about 3.6 kilograms and 300 millimeters. Just yesterday, or the day before, yesterday, our field technician Felix captured one that was over 6 kilograms. That was really, really big. And so they get pretty big. And in this one, we've learned a little bit more because we've found 2 females nesting in May 2022. So this year, in the morning of May 2022. So this year, we're hoping to observe more nesting females. And also seeing this female has allowed us to understand a little bit more about when Trachemys are nesting. And for example, in nebulosa, we haven't observed them nesting, but I've gone and looked at on the beaches along the Cuchunaki River in September, October, November, and I can see little turtle tracks coming out of the sand. And I've got a turtle dog, and we've gone there. We've tried to find the nest, but not yet. Figuring out nesting in turtles is one of the hardest things in herpetology to do. They're really tricky to figure out the nesting patterns, unless you're John Iverson. So the main concern for Trachemys yaqui that we started observing in the Taggert Reserve is the drought, because when we first started sampling in 2022, 2023, we had good sampling success. In 2024, it was still pretty good, but in 2025, the dam nearly dried. The sampling success went to the floor, and we observed dead turtles and fish in 2025. And so, over the years from the drought, the sampling success really decreased. And this year, although I didn't include these data yet, we had 3 weeks of trapping, and we've trapped, I think, 5 turtles, and our abundance estimate, our turtles per trap night, would be less than 0.1 right now on the year. So this drought has become a major theme in the Southeast, and I'm sure a lot of you know about it. We're in this extreme drought, and the driest period since the 1500s, and the second driest since 800 CE, and it threatens not only organisms but also ecosystems and ecosystems in the region. And there's actually a rain gauge near Alamos, in the town before you get to Alamos, in Minas Nuevas, that's been recording since 1927, and from 2020 to 2024 was the driest 5-year period since it started recording. A lot of these organisms are adapted. A year with 300 millimeters of rain isn't something that hasn't happened in the past 100 years, but 3 or 4 in a row is something that hasn't happened in the past 100 years, and it is starting to produce observable effects. And we first noticed this in 2020. We were studying the Alamos mud turtle, which we'll talk about for a second. So Sonora alamosi is also endemic on the Pacific coast, found in Sonora and Sinaloa. And 2019, we started studying them. We had a good rain year. We put radios on individuals, on 5 individuals. We had good trapping success. But the next year, we didn't get any rain. We didn't get enough rain to fill up any of the ponds, so they didn't get water that year. They estimated for 18 months, and 2 of those turtles died going into 2021. In April and May, 2 of them died of parent desiccation, and one of them made it all the way to the pond, but died right at the edge of the pond. And using a sampling effort, and these populations, we do have more robust population data. Capture rate decreased by more than 30%. So these droughts have also affected species like Sonora alamosi. But one thing about them is that they can reproduce, and they can get to adult size in 5-6 years, and turn over in a population much faster. They only need a few good years to be able to rebound. And this is also something that the Mokuseri Dam also nearly dried in 2025. So Mokuseri Dam. So if you see where this river comes out, that's actually the type locality of Trachemys yaqui. And this is a picture of the dam at like 50-60% capacity. And in June 2025, it was at 0.7% capacity. And it was there was a regional crisis. The water table fell fell really low. Most of the wells in the community were dry. Some of the city wells were dry. And the farmers were having trouble with their cows at getting water. A lot of them were selling their cows. In our house there, we were getting a truckload of water, 1 truckload of water per week. And it was just the thing to talk about in June 2025 was how hard the drought was. And the last time you took a shower. And so, and also, although Trachemys aren't found in the pine oak forest, in the dry season in 2025, I was living in Puerto Vallarta, and my field technician and a colleague of mine that were going up to the Santa Barbara, which is pine oak forest, they were telling me, pines are drying, oaks are drying, and me, being naive, the rainy season's going to happen. Don't be so negative. They're gonna come back. But after spending, a couple of months there, we had good rain last year, luckily. Most of the trees didn't come back, and about 30% of all the oaks are completely dry. In this picture, you can see this patch of all these dry oaks, and this is already after we've had 400 millimeters of rain. or 300 millimeters rain, we had a really good year. You see all these dry pines, and you see this entire swaths of dry pines across the base of the mountains. So this is just another reflection in the region of how this drought is affecting the area. So we're really hoping that we don't repeat the 5 years of drought that we had the 5 previous years. We had a good year last year. We've had good winter rains, so we're optimistic that we'll have a few good years. So conclusion on Trachemys yaqui, we only have data on two populations, and the majority from one single population, but the drought seems like it could present a serious threat to Trachemys yaqui. So sampling throughout the range is desperately needed because the drought has been affecting throughout this entire range. If you go up in the north, if you drive through Cananea right now, I think they got even hit harder. It seems like, they might have lost 80% of their oaks and junipers. I think there's junipers there as well. Just the hillsides there look completely dry. It was a very depressing season to live through that and imagine what the next few years will be like with the more hot adapted species invading the beautiful pine oak forest that I love to spend time in. And so sampling is desperately needed, and we currently, we have a grant to go to these sites throughout northern Mexico. I actually came up to Tucson to pick up new traps for this sampling effort. So some concluding remarks. Trachemys species in general have been overlooked throughout Mexico, and they're facing, the main threats they face are the invasive species from this, pet trade of these little baby, sliders. Consumption, we observed that in Sinaloa, but in the southeast, Richard Vogt mentions, populations he visited in the 70s, early 80s, and goes back to the 90s, and he seemed to suggest that some of them were decimated by consumption and drought. And our studies in northeast Mexico or northwest Mexico, I made a mistake there, suggested the perception that most species are stable may be misguided. So, we shouldn't assume that any of these species are stable. And, we tend to not worry much about sliders and, focus on more charismatic species like box turtles, like species really small distributions, or big charismatic species like the Central American river turtle or tortoises. But we also need to think about the sliders. So, thank you so much to Tucson Ecological Society for inviting me. And there's a lot of collaborators, and especially the local reserves that allow us to work on their properties. And follow us on our Facebook to follow our work. And I brought some pamphlets we have that the Turtles of Sonora that we'll pass around. I already gave some of them out.
Yeah, go.
Thank you, Taggert.
I don't know if there's questions.
Really great. Where yaquia and nebulosa are sympatric, you noticed any hybrids between those two?
And in the individuals we observed, we didn't notice that there could be hybridization.
Okay.
And that's also another exciting thing about finding this locality, because maybe finding where they overlap could, and for example, in those sites, if they don't hybridize and there is reproductive isolation, it could help explain how Trachemys yaqui became isolated from Trachemys ornata.
Yeah.
Because there's not a clear geographic barrier along the coast that would separate species.
So that's related to my next sort of observation. You feel like nebulosa is from Baja and cross the ocean to the mainland. Is that what you're saying? That's really interesting. There's see that with bujum trees.
I was going to mention that example.
Yeah. Okay. Yeah. So and yeah, very, very interesting. And there's a whole geogenomic project that's trying to elucidate all of those questions.
I should talk to them.
Yeah.
Yeah.
Do you have a sense of how much these turtles are moving up and down the water courses?
Yeah, and that's one complication of the Cuchuhaqui River, and why we don't have the same comparable data from one year to the next, like we do in the Titaciosa Reserve, where we know they don't move. Because, for example, in the downstream site, the turtles seem to be pretty consistent. We find recaptures from previous years in there, but also if we get a big flood year, we'll find we'll have a good amount of new turtles. Like this year, for example, we had a big flood year last year. I didn't include the data from this year, but we got a lot of new turtles there this year. So it does seem like they were probably way upriver where we can't sample, and now they're down there, or they came up from downstream. And for example, this last season on the upper reaches, the midstream tributary and the upper stream, we barely got turtles there this year. And I don't think we've captured any turtles there this year or last season. And I don't think it's because they went extinct there. I think it's because we had this really big rain year. Some of those canyons and they're very difficult canyons to navigate. They're like sheer rocks, and you can only get to them in certain places. And one of them like filled up with sand, and it seems like if there's a canyon, it's probably further up where we can't access. And so there's these complications with that waterway where we do think that they move, but we've never gotten a recapture from downstream at the midstream site or the midstream at the upstream site, and vice versa. So they probably do move around. We just don't know how much yet.
In terms of moving around, I keep thinking that these sliders can get out of the water and cross country. Doesn't have to be dispersally in the water.
Yeah, not necessarily in the water, and yeah, that for example, the artificial pond that we sample way south of, both the ponds one of the southeast ponds where we find both species, that's like 2 kilometers from the river, so it's not really hard to think that a female, got out while she was laying eggs, and she got, she ended up there, but the furthest southern ponds are 8 kilometers from the Rio Fuerte River. So, it is interesting to think about how they got all the way over there. And, someone one time was going down one of the country roads and found a nebulosa crossing the road kilometers away from the Cuchahaque River during nesting season. So during the wet season, they can get out on the bank and walk. But also during nesting season, we know females move, and there's a lot of open questions about how much and when they move outside of the water.
So I'm trying to figure out there is not a geographic barrier or something that separates. It's so distinct, these two distributions. Is it a watershed so that the Fuerte watershed's got nebulosa hiltoni?
Yeah, and Cuchahaque is a tributary of the Rio Fuerte.
Right. So the Rio Mayo has the yaquia.
Well, the Rio Mayo, the Yaqui River, and some other river systems in northern Mexico have Trachemys Yaqui.
So there's a geographic separation with the river system, not the distribution of the birds.
Yeah.
So both species have crossed their respective watersheds. It sounds like.
We haven't gotten Trachemys Yaqui in the Cuchahaque River.
Okay.
But it's close.
It's close.
Yeah.
Those are the southernmost records that I know.
Yeah.
Jeff Lovitch has I think a couple of questions. Oh, Jeff Seminoff, sorry, and then Lovitch. I'll just read it for the folks on Zoom. Speaking of local consumption of Trachemys in Sinaloa, was there any evidence of freshwater turtle meat and sea turtle meat mingling in the same illegal supply lines, or consumption of both in the same social circles? Also, any intersection of freshwater turtle meat consumption and narco trade?
Well, we didn't see any crossover between sea turtles and the freshwater turtles, but I would imagine that the people who eat turtles would eat sea turtles or freshwater turtles. But we were asking about the freshwater turtles, and no intersection with freshwater turtle meat and consumption and narco trafficking. We did have some people with radios checking us out, but no intersection. They seem to not really care about turtles in general. At least there, maybe down in Jalisco, it's a different thing.
Thank you. I'm guessing that's Jeff Lovitch. Nice work, Taggert. I didn't see any photos in your talk of melanistic individuals. Generally, that trait is seen in Trachemys in the US, not those farther south. But melanism has been reported in T. yaquia, did you observe any melanistic sliders in your samples?
We haven't observed any melanistic sliders. We've observed older individuals that don't have any color anymore, and Trachemys nebulosa compared to others is more melanistic in general. Just the species in general, they're much darker in general, and yeah, I haven't observed melanism in that cesspool of a pond in Puerto Vallarta. There's a ton of individuals with melanism, especially the scripta that sometimes don't even look like scripta. But yeah, in Sonora we haven't observed that melanism except in very old individuals that could have just lost all their color because they're so old. And she had a question.
I had a question kind of related to that. Thank you. Great talk, by the way. You showed a photo of a hybrid between nebulosa and the petrade turtle. And it was quite large. So I wondered if it was like a fairly old individual or if you know.
Oh, it's Trachemys or not? That was a that I took in Puerto Vallarta.
Okay.
And it was a juvenile. It looks like this big.
Okay.
And so for Ornata, it's still, 3 or 4 year turtle. It's still pretty young. But for Trachemys scripta, it is big. And some of those hybrids that we get in that pool in Puerto Vallarta do get much bigger than Trachemys scripta elegans.
So do you know how long that release hybridization might have been happening, or can you tell from the age of the turtles that you get?
Say that again.
Like, can you tell how long the hybridization might have been happening by the size and age of the hybrids you've captured?
If we really spent time measuring the turtle, well, we measure the turtles and we mark them, but we don't take the photos of all individuals. And when in that pond in Puerto Vallarta where we sample them, we get so many hybrids. It's like so overwhelming to be there all morning. We'll get like 150 hybrids, and we're marking all of them with triangular files that start getting gummed up, and it's just a ton of work to process all those turtles. But I do suspect that we could figure out stuff like seeing the biggest hybrid individuals, and there is things we could learn by looking at the age classes and the growth of younger individuals, but we haven't spent time. And one thing there too, if it's and we've classified turtles in that pond as Trachemys scripta elegans or hybrid, and we don't say it's a hybrid or red-eared slider. We just it's another hybrid or elegans, and because our plan is to take them all out, but we just haven't been able to do that.
That's the hard. What's maybe I missed this. How much hybridization are you noticing in Northwest, like with yaquia and or nebulosa?
So I haven't observed. Well, we observed that one hybrid individual of Trachemys nebulosa, and on iNaturalist, like for example, it appears that there could be some hybridization in Hermosillo, but it's really hard to tell from those photos. And so directly observing hybridization, we haven't seen it anywhere in Sonora. We haven't seen it anywhere in Sinaloa. And in Los Mochis, there's this public pond at I don't know if it's a botanical garden or park in Los Mochis, and they got this pond where you go and see nebulosa, and there's a ton of nebulosa. And I couldn't see any clear hybrids there, but there is scripta elegans, there is Trachemys ornata, there's some map turtles, and I didn't notice. And if I did, if I'm remembering seeing hybrids, it would have probably been Trachemys elegans with ornata. And when there's 4 species in a pond, it's really hard to figure out what's a hybrid.
Yeah. Okay.
But and one interesting point I didn't pull up there is in San Jose del Cabo, where we didn't sample. I'd been there a few years before and looked for turtles in the estuary, but when I saw them, I could see turtles there. They didn't look like nebulosa. It seemed like they could be Trachemys venusta or nata. And we didn't sample that site, but there's hundreds of observations from San Jose del Cabo, and they're all Trachemys ornata. So it's not clear if those were introduced pets or reintroductions, or if it's a natural migration of ornata coming from the mainland to the southern tip of the peninsula. But a lot more, genetic work would need to be done to figure that out.
That's so weird. So the sister species to nebulosa is Meliclenus?
I don't think so. I think from understanding that paper, I'm not a genetics person. It comes up as Malaclemys because it retained this weird part of the genome that makes it difficult to distinguish from other species.
That's not that they're not related, it's just a weird quirk of the genome. Okay.
And other papers, like there's Bradley Schaefer's lab came out with the phylogeny of all turtles, and I think that was, he was only nuclear markers. I remember him mentioning that peculiarity of Trachemys nebulosa, and they come out sister to like the Q in sliders or something like that. Yeah.
So nothing further south. That's right next door.
No.
Weird.
Yeah, they're not part of the Gray Eye Venusta ornata group. Yeah.
It should be southwestern Mexico.
Yeah. Okay. The Gray Eye.
Okay. Well, thank you, Taggert.
No, thank you.
Yeah. Any other questions? All right. Okay, yes. That's very good. All right, thank you, folks, on Zoom. So let's see, you want to go ahead and
I don't know. I'm sorry.
And,